My name is Princess PASILBAS, and before I begin my lecture, I have a few pictures that I want you to see. So here I have pictures of bread, rice, and potatoes. Now what do you think these three have in common? So, these three have starch in common. Now what do you think happens when these foods enter our mouth? Do you think it turns to sugar right away? Well, the answer to that is, even before these foods reach your stomach, a chemical change begins in the mouth, where starch is broken down into smaller sugars. Now, have you ever noticed how plain rice and bread taste sweeter the longer you chew it? That's not your imagination. That's maltose forming right in your mouth. Now, understanding about maltose and how it's formed is important, but not just in theory, but for future lab work. So that's why I'll be discussing this lesson, and I hope that you will understand as we go through it. So let's get started. Before we begin, here are the different learning outcomes for today's lesson. So by the end of this, you should be able to first, define maltose as a disaccharide. Next, describe the structure of maltose, including its monosaccharide components, type of glycosidic bond, and its key properties. Third, discuss the nutritional significance of maltose in the human body. And fourth, identify the enzymes involved in starch digestion and describe their specific roles. Lastly, explain the process of starch digestion leading to the formation of maltose. These outcomes will guide what we focus on today, so keep them in mind as we go through the topic. So let's begin by defining what maltose is. Now, maltose is a disaccharide, which means it's made up of two simple sugars. Now, in the case of maltose, it's made up of two glucose units, and these glucose units are chemically bonded together. Now, how do we get maltose? Now, we get maltose when starch begins to break down. And another thing about maltose is that it's also commonly known as malt sugar. Now, this sugar is sweet, but it's not as sweet as sucrose. So in short, maltose is equals to 2 glucose units, and it's a disaccharide type of carbohydrate. Here I provided you guys a picture of the structure of maltose to help you visualize what I just explained. So each maltose molecule is made up of 2 glucose units, specifically an alpha-D-glucose unit. And these glucose units are what we call the monosaccharides, the simplest forms of sugar. Now when these 2 glucose units join together, they form a glycosidic bond. So that's what I meant earlier by chemically bonded together. So specifically in maltose, they have a alpha 1,2,4 glycosidic bond. Now why is it alpha? Why is it 1,2,4? So alpha because, as you can see, you have to check whether it's an alpha bond or a beta bond. You have to check the placement of the hydroxyl group of your anomeric carbon. Your anomeric carbon is the carbon after that oxygen. You see that oxygen there, the O? So after that, that's to its right, that's your anomeric carbon. And as you can see in the structure, the hydroxyl group of that anomeric carbon has its hydroxyl group below. So that's why we classify it as an alpha bond. So for both glucose units, it has its anomeric carbon has its hydroxyl group below. And why is it 1,2,4? So 1, because in the first glucose units, the glycosidic bond is linked to the first carbon of the glucose unit. And why is it 4? So because in the second glucose unit, the bond is connected to the fourth carbon of the glucose unit. So that's why it has a alpha 1,2,4 glycosidic bond. Now, how is this bond formed? It's formed through a condensation reaction. Now, meaning what happens is that a water molecule is released and the two sugars are joined together. So that's why the final products of this reaction is maltose plus water. And another thing is that the alpha bond actually plays a key role because specific enzymes recognize this bond and act on it during digestion. So before we move on to the next segment of our discussion, I have a few questions to ask you. So first, what do you think are the two simple sugars that make up maltose? You can pause the video to think for an answer. So the answer is 2 alpha-D-glucose units. Next, is the bond connecting the glucose units an alpha or a beta bond? So the answer is, it's an alpha bond, specifically an alpha 1,2,4 bond. We've seen what maltose looks like, we've understood how it's formed, what it's made up of. Let's now move on to its different properties, starting with its solubility. Now, maltose dissolves readily in water due to its polar hydroxyl groups, which means it can move to the bloodstream and into cells. Next, its taste. Now, maltose is sweet, but it's not as sweet as sucrose or table sugar. Third, its sugar type. Now, as we all know, maltose is a disaccharide because it's composed of 2 glucose units. And lastly, reducing property. Maltose is a reducing sugar because its maltose unit, its second glucose unit, has a free anomeric carbon that can open up to form an aldehyde group. And this aldehyde group can participate in chemical reactions, which is important in diagnostic tests like your Velez test. So those are the few properties of maltose. Let's now move on to the nutritional significance of maltose. Nutritional significance of maltose, meaning its benefits to our body. So maltose can act as a quick energy source. Why? Because maltose, when broken down, produces two glucose molecules. And this glucose molecules is then rapidly absorbed and used by our body for energy production, especially during physical activity or when our blood sugar is low. So this is common in sprouted grains like barley and wheat. It's also found in fermented food like beer, miso, and soy sauce. And it's also formed during digestion of starch-rich food like your bread, rice, and potato. While maltose is a helpful energy source, excessive intake of it can lead to spiking up your blood sugar. So that's why it's important to eat in moderation. So now that we've covered its nutritional significance, let's now move on to the different enzymes that are involved in starch digestion and its specific roles. Let's now talk about the different enzymes involved in starch digestion. So starting with our salivary amylase, or your ptyalin, which is located in the mouth. So when we chew our food, so when saliva comes in contact, it has an enzyme called the salivary amylase, or your ptyalin, and what it does is that it breaks down the polysaccharide into disaccharide. So your starch to maltose. Next, your pancreatic amylase, which is released by your pancreas. So what it does is that it continues the breakdown of starch. And lastly, your maltase and your sucroisomaltase. These are brush border enzymes of your small intestine. So what it does is that your maltose is converted to glucose by your maltase, and your sucroisomaltase converts your alpha limit dextrins to glucose also. So those are the different enzymes involved in starch digestion. Now let's go over the whole process of starch digestion. So I've now introduced the different enzymes involved in starch digestion, right? So now here I'll discuss the whole process of starch digestion, so we can see how the enzymes work and the final product for this entire process. So how does this exactly start? So first, starch digestion begins in the mouth. So when your food comes in contact with your saliva, when you chew it, your saliva has an enzyme called salivary amylase, or ptyalin. And what that does is that it breaks down your starch to maltose. However, that's just the initial stage of digestion. Second, your food enters the stomach. However, the acidic pH of the stomach inactivates your salivary amylase. Why? Because the acidity of the stomach is about 1.5 to 3, which then denatures the enzyme, leading it to its inactivity. And your salivary amylase works best at slightly acidic to neutral conditions, so about 6 to 7 pH level. So that's why in this area, no starch digestion happens. Third, partially digested food now enters the small intestines, and your pancreas will release an enzyme called pancreatic amylase. Now what this does is that it converts the remaining starch to maltose, maltotriose, and alpha limit dextrins. And fourth, we now reach the intestinal brush border. So the intestinal brush border has enzymes we call the maltase and the sucroisomaltase. Your maltase will now break down your maltose to the two glucose molecules, and your sucroisomaltase will then break down your alpha limit dextrins to glucose. And these glucose molecules will now be absorbed into the intestinal lining, into the bloodstream, where it will be transported to the liver and the body cells. So the glucose will be transported to these areas for it to be used as immediate energy, or it could be stored as glycogen for future purposes. So that's it for the entire process of starch digestion. So it goes starch to maltose and then to glucose. So the final product of this is glucose, and your glucose will be used for energy production. So now we finished the discussion. We've covered the definition of maltose, its structure and components, properties, nutritional significance, and the process of starch digestion. Now, it's your turn to apply what you've learned. So I'll be giving you guys a simplified diagram of starch digestion, and what I want you to do is pause the video and write down a short answer as to how starch from food becomes glucose in our body. So you should include key terms that I've mentioned in my discussion. So if you listened, you'll know. So you can do this solo or with a pair, whatever works for you. So pause now, and when you're ready, press play to hear my answer. So a simplified answer to that would be starch is broken down to maltose by your amylases, such as your salivary and your pancreatic. And your maltose is broken down to glucose by your maltase. And that's it. If you got it right, then good job to you. So before we wrap up the video, I'd like to give a quick recap again of everything I've discussed in case you guys forgot. So again, your maltose is a disaccharide which is composed of 2 glucose units linked by an alpha glycosidic bond, which is alpha 1,2,4 glycosidic bond. And your amylases, your salivary and pancreatic, convert your starch to maltose, and your maltase and your sucroisomaltase converts your maltose to glucose, and your sucroisomaltase converts your alpha limit dextrins to glucose. And this glucose will then be absorbed in the intestinal lining into the bloodstream, where it will be transported to the liver for it will be used for immediate energy, or it could be stored as glycogen. So understanding this process and the whole discussion basically is actually important, because this will help us see how the food we eat becomes energy, and this is actually an idea that will actually come up in future lab work and future discussions. So I hope you guys listened, and I hope you guys understood. So thank you for listening, and bye-bye.